Mutations and sequencing. So mutations are changes in the genetic sequence that may or may not alter the protein. They're detected in several ways: biochemical methods and nucleic acid methods. Biochemical methods would be immunoassays, high-performance liquid chromatography, or mass spectrometry. So the biochemical methods, if we look at immunoassays, they're performed with defined antibodies that detect the mutant or wild-type proteins. Immunohistochemistry is a cytology method to identify normal and mutant proteins in tissue sections. This can determine if protein levels are upregulated or downregulated in any type of sample or even in tissues. And antibodies detect specific wild-type or mutant proteins. So we're not detecting a mutation directly on the sequence of DNA. We're detecting if there is a mutation, then it would produce a mutated protein, and we'd be able to pick up on that mutated protein. So this would be like the phenotypic expression of that mutation, not the genotypic analysis of the DNA. Similar thing here. So we have high performance liquid chromatography, uses solid and liquid phases to separate particles by size, charge, or chemical characteristics. DNA is amplified using PCR. PCR product is double-stranded, and then it's partially denatured. If a mutation exists, then the mismatch may be retained in the column longer compared to the wild-type DNA. So we amplify a particular region that we're interested in. So let's say we know that there's a mutation that could exist in the population in this particular gene. Well, we're going to amplify that gene, and then we kind of partially denature it or separate those strands, and then the strands are going to want to come back together. But if a mutation exists, the mismatch may be snagged up in the column, and it may take longer to get that through than the wild-type DNA. So then we can determine that, yep, there's something wrong with this piece of DNA. In gas chromatography, samples are vaporized and separated into a gas. If a mutation changes the amino acid sequence, then it changes the mass of the protein, which can be detected. This is another one where we're looking for the product, the protein product, not necessarily the gene sequence. MALDI. So you may be familiar with MALDI from microbiology, where we use MALDI in order to basically fractionate all of the ribosomal proteins and then get a read on those ribosomal proteins. When we use a laser to deionize and absorb those proteins, we detect them, and each different type of protein is going to produce a different signal, and each different microorganism then has its characteristic signal. We take our unknown organisms and compare them to the database of the known organisms in order to come up with an identity of that organism. So we can do something similar here. Matrix-assisted laser desorption or ionization mass spectrometry is used in molecular separations. So PCR amplifies a region of DNA. A primer is used on the target. That primer is going to add bases up until the suspected mutation site. Then it's going to add the base that is complementary to the next base, the mutated base. Depending on which base is added, a different mass can be detected. So it'll detect the primer plus that additional nucleotide. And because this is very sensitive, each different nucleotide that is added is going to be detected. So if an A is added, then it's going to pick it up as a particular signal, and that is going to be different than if a G were to be added. So this shows the PCR amplification, and then the primer, when it's added, it's going to add all the way up until the mutated base, and then this special nucleotide is added at that region. Now when it's added, whatever base is added can be detected by MALDI-TOF. Nucleic acid-based detection methods, there's hybridization-based, single-strand conformational polymorphism, melt curves, array technology. There's sequencing, polymerization bases, or based, sequence-specific polymerase chain reaction, allelic discrimination. And then there's cleavage-based, restriction fragment like length polymorphism or nuclease cleavage. So starting with single-strand conformation polymorphism, scans several hundred base pairs, and based on intrastrand folding. You PCR amplify a region, you denature it into single strands. The strands are going to fold based on the sequence within the strand. One base difference will affect the folding. So folded single strands or conformers can be resolved by size and shape on gel electrophoresis. This does have strict temperature requirements because the folding is temperature dependent, and keeping the strands separated from one another is also temperature dependent. So single strand conformation polymorphism, you can see if you run it out on a gel, those are the normal ones, those would be mutant, and those would be normal and mutant together. Again, what we're doing here is we are amplifying a region of DNA. The amplified region is going to be double-stranded DNA. So then we heat it up to a particular temperature where it's going to separate the strands into single strands, and those single strands are going to then make a folding pattern. So they're going to be single strands that fold, and different bases are going to cause it to fold in different ways. And once it's folded, it's going to migrate through the gel differently. Melt curve analysis is based on the sequence effect of Tm, or melting temperature. We talked briefly about melting temperature when we talked about PCR. Melting temperature is dependent on the concentration of G's and C's because we have two hydrogen bonds that hold together the bases A and T, those complementary bases, and then the bases C and G are held together by 3 bonds. So if there's more C's and G pairs in a particular sequence, we're going to have a much higher melting temperature. The longer the sequence is also going to impact the melting temperature. And then if we have mismatched bases, they aren't going to bond to each other, so that can decrease the melting temperature. So base alterations have to be significant enough in order to impact the melting temperature for this to work. It can be performed with or without probes. It requires dyes specific to double-stranded DNA, like ethidium bromide or cybergreen, also performed with fluorescent resonance energy transfer, FRET probes. You PCR amplify the gene of interest. Then you slowly increase the temperature to melt the DNA into single strands, kind of similar to what we did in the previous method. But then you're going to add fluorescent probes that only bind to double-stranded DNA, so the fluorescent signal fades once the single strands are formed. Then you're going to use detection instrument software, which may convert the melt curve to a derivative of fluorescence, the speed of drop versus temperature. Every sequence has a characteristic melting point. If it melts faster or slower, then we know that the sequence is not wild type. So here you can see higher melting temperature would be homozygous, lower would be homozygous mutant, and then a hybrid would be somewhere in the middle. Array technology. Arrays are reverse dot plot methods. They're used to investigate multiple genomic sites simultaneously. They're grids of microscopic spots. Each spot contains a specific short, single-stranded DNA probe. The specimen DNA is often PCR amplified and then labeled and hybridized to immobilized probes. If they stick, then their sequence match. So I don't have a picture of this, but basically if you picture a little card and you picture all of these dots in a row, in rows on the card, and every one of those dots has a very specific single-stranded DNA probe. Now we're going to take our specimen DNA and we label it. So we're going to put some type of amplified or amplification signal on our specimen DNA. Then we're going to inject our specimen DNA into all of these different grids, all of these different spots on the grid. And if they're a match, they'll stick and produce a signal in that spot. And then that signal can be detected. So this allows us to detect a lot of different, very specific sequences that are present. Microarray technologies are listed here, so there's several different technologies that exist. You can review this table to make note of the differences. Sequence-specific primer PCR, SSP PCR. The PCR primer extension requires that the 3' base of the primer is complementary to the template. So a normal template, the PCR primer is going to go perfectly. In a mutant template, the PCR primer is not going to stick at a particular region, and no amplification would happen. The primer design is used to detect mutations in the DNA. The generation of a PCR product indicates the presence of the tested mutation or polymorphism in the template. Allelic discrimination uses 2 fluorescently labeled probes. One binds to wild type and one binds to a specific mutation. Quantitative PCR technology generates a color or signal for the mutant or the normal sequence, and this is performed on real-time PCR instruments. Restriction fragment length polymorphism. Restriction enzymes are designed to cut at specific sequences. If you have a mutation in the cut sequence, it's going to cut the DNA at different lengths. So the restriction enzyme site recognition detects the presence of sequence changes. For example, a G to A change creates an E-core 1 site, and that's going to create different cut bands that you can then resolve on a gel. Invader technology. Mutation detection with proprietary enzyme. The sample is mixed with 2 probes and enzyme. One probe invades the double-stranded DNA and binds to it and keeps those strands separated and binds just before the mutation. The other overlaps and binds over the mutation. Then you have enzyme cleavage of the probe test sample hybrid. It will yield fluorescent signal. The signal will only occur if the probe and test sample sequence are complementary. So this shows an image of this. So we have our invader probe that binds, and that forms this flap. And there is an overlap at this base here. So mutant test DNA is leaved here. We have cleavage here and no cleavage here. Point mutations, single or a few nucleotide changes, not detectable by cytogenetic methods, detected at the DNA sequence level. These are how mutations are named. So the base position, the original base, and the replacement. Here's the original amino acid position, the amino acid position, and the replacement. You can see a deletion is listed like this, and insertion is listed like this. Termination listed here. Point mutations do not always have a phenotypic effect. So here, there's no mutation, so there's no effect. Here we have a point mutation where the G was replaced by a T. It's still going to produce the same amino acid sequence, so that's called a silent mutation. Here you can see the G was, or the A replaced the G. This did cause an amino acid replacement, but the amino acid is pretty close in structure, so it's called a conservative mutation. The protein's still going to fold relatively in the same way. Here we replaced the A with a C. Here we've got a non-conservative, the mutation. It's an entirely new amino acid with a new structure. Here we replaced the C with a G. Now we've created a stop codon or a termination sequence. That's known as a nonsense mutation. So it cuts the amino acid chain short. So we will not create a full polypeptide here. And then here is a frame shift mutation. So we have added a base in here and shifted the rest of the reading frame down. So anything distal to that insertion will be the incorrect amino acid sequence. So now we have added different amino acids to the sequence. So the summary is here that you can review, and we're going to go on to sequencing. So there's different types of sequencing methods that exist: the Maxim-Gilbert, Sanger chain termination, pyrosequencing, and next generation sequencing. With these different sequencing methods, the goal is to get the order of bases on a particular strand of DNA. The Maxim-Gilbert sequencing was performed by chain breakage at specific nucleotides. Chemicals cut the DNA at specific bases, and then we can evaluate the strands on a gel. So sequencing gels were read from the bottom to the top, and it's based on that chain breakage by the chemicals. This procedure is no longer performed. The chain termination or Sanger sequencing is a modified DNA replication reaction. Growing chains are terminated by dideoxynucleotides, DDNTPs. They have a slightly different structure, which prevents them from adding more bases after them. The 3' OH group is necessary for the formation of the phosphodiester bond, and it's missing, so that's going to terminate the extension. So a sequence reaction mix includes labeled primer and template. So we have labeled primer, and then the template DNA and the area to be sequenced. The primer is going to stick to the template, and then we're going to try to make copies of the DNA sequence. While we're making the copies, we have 4 different dideoxynucleotides: the dideoxy A, T, C, and G. And those are added separately to each of 4 tubes. So we have the DDATP, the DDC, the DDG, the DDT. So that's going to add A, C, G, and T bases. In order to figure out the sequence of a strand of DNA, the primer is extended until a DNTP is encountered. The chain will end with the incorporation of the DDNTP. With the proper DNTP-DDNTP ratio, the chain will terminate through the length of the template at every single base. All terminated chains will end in the DDNTP added to that reaction. The collection of fragments is a sequencing ladder. The resulting terminated chains are resolved by electrophoresis. Fragments from each of the 4 tubes are placed in separate gel lanes, and then it's read from the bottom to the top. So if we had a strand of DNA, so we have this strand of DNA, what we would do is upstream of this strand there would be a template region, and we would have a primer. The primer would bind, and after the primer bound, we would start making copies of the DNA. Now, as we're making copies, so we're replicating the DNA strand, and as we're making this replicated strand, we are going to randomly either incorporate a regular amino acid, or a regular nucleotide, or we're going to be incorporating one of those dideoxynucleotides. If we incorporate a regular nucleotide, and let's say we only have A's that we can incorporate in this tube, it's going to make a copy, and it's going to incorporate an A, and then it's going to incorporate another A and another A, and then it's going to incorporate the dideoxy A. So we can copy this strand. We're going to have A, G, C, G, T, C, C, T, T, and then if as soon as we incorporate one of those dideoxy bases, it's going to stop. It's not going to be able to continue. So then we're going to run the sequence again. We're going to start with the primer. We're going to start making copies of this. And now we're going to incorporate it right at the beginning. Then we're going to do another reaction, and we're going to make the copy of the genetic material, and then we're going to incorporate it right here. So as soon as it's incorporated, we don't make any more copies. After all of this is done, we run it out on a gel. So this A has been incorporated first. We know that this is the first letter in the amino acid because, in the strand of DNA, because it's at the bottom of our gel. This is going to be the smallest fragment. It's one base pair in length, so plus the primer. So it has this smallest fragment. Now the next one up would be this one, the G, where we had the G incorporated, because when we had the A and the G, it would then be the primer plus the A and the G sequence. And then we kind of just read this going all the way up. Sometimes with very large sequences, it's difficult to read near the top. So we need really long gels, and those really long gels need to be run for a long time and separated under very slow voltage. So that's one of the limitations in like the standard Sanger sequencing method. Advances in sequencing technology, we have cycle sequencing. So we used to do this all by hand, but cycle sequencing basically is performed in a thermal cycler, just like in PCR. It does require a heat-stable DNA polymerase. Then we advance to fluorescent dyes. Fluorescent dyes are multicyclic molecules that absorb and emit fluorescent light at specific wavelengths. Examples are fluorescent rhodamine derivatives. For sequencing applications, these molecules can be covalently attached to nucleotides. In dye terminator sequencing, the fluorescent dye molecules are covalently attached to the dideoxy nucleotides, labeling the sequencing ladder at the 3' ends of the chains. The distinct dye or color is used for each of 4 nucleotides. So a different color is used for A, T, C, and G. Because the terminating nucleotides can be distinguished by color, all 4 reactions can be performed in a single tube. So you don't have to use the different tubes because each one of those dideoxy nucleotides is a different color. So we're going to put them all in the same tube, and then we can have the primer, the template strand, and it's going to make copies of this. And then we can run it out on a gel or in capillary electrophoresis. Capillary electrophoresis is a much more efficient system, and it's able to separate the bands a lot more easily with having a much smaller gel. So with capillary electrophoresis, it allows us to sequence much larger fragments because they go through these little capillary tubes instead of having to be run out on a very large gel. The DNA ladder is read on an electrophenogram. So if we do this type of sequencing, then we pick up on the color changes, and it's detected using this. Sequence analysis software provides analyzed sequence in text and electropherogram form. Peak patterns reflect mutations or sequence changes. Alternative sequencing methods, pyrosequencing. So pyrosequencing is based on the generation of a light signal through the release of pyrophosphate upon nucleotide addition. Pyrophosphate used to generate ATP from adenosine phosphosulfate. ATP and luciferase generate light by conversion of luciferin to oxyluciferin. Each nucleotide is added in turn. Only one of the 4 will generate a light signal. The remaining nucleotides are removed enzymatically. The light signal is recorded on a pyogram. So imagine sequencing a short template strand with the following sequence, A, G, C, T. In the first cycle, we're going to add A. Since the first base is T, no light will be emitted. Cycle 2, we add the T. The base is complementary, so light is emitted. Cycle 3, we add the C. Since the second base is G, no light will be emitted. The cycle 4, we add G. Light is emitted as it matches the template. The burst of light after adding T and G will allow you to sequence or tell that the sequence is AG. DNA template is immobilized and primer is added. Nucleotides are sequentially added. Pyrophosphate is released and each nucleotide is incorporated. Sulfurylase and luciferase convert pyrophosphate into light. Light is detected and the sequence is constructed. So this is a diagram. You can pause the video and review this. The Human Genome Project. The first sequence of the human genome by Sanger sequencing took 10 years and cost $500 million. Complete sequence and comparative analysis of each chromosome followed. By 2006, all of the chromosomes were sequenced. The human DNA sequence, 2.91 gigabase pairs, 36 to 62%, GC, or G plus C, less than 30,000 genes. Average size is about 27 kilobase pairs. Less than 2% of the genome is open reading frame. 42% of the open reading frames have unknown function. Genes are not evenly distributed within or among chromosomes. Difficult areas to sequence include centromeres, polymorphic regions, and major histocompatibility complex. The 1,000 Genomes Project. The 1,000 Genomes Project was funded by the National Human Genome Research Institution. It was intended to identify variations in the human genome. So the original human genome that was sequenced was a white male. And unfortunately, white males don't contain all of the genes for all of the other races, ethnicities, genders. We can't just base it off of one individual. So we got the genotypes from more than 2,500 individuals, and they've now been made available. These databases are now used in bioinformatics to identify polymorphisms. So polymorphisms have to exist in at least 1 to 2% of the population. So 1 to 2% of the 25,000 individuals should have this in their genome for it to be considered a polymorphism. Next generation sequencing technologies. So ion conductance, reversible dye terminator, pyrosequencing, and single molecule long range. Next generation sequencing involves generating a library, sequencing the library, and then analyzing the sequence. A sequencing library is the DNA to be sequenced. Microbial populations, whole genomes, exomes, specific genes or regions, standard or paired end, RNA, and complementary DNA. Fragment DNA to several hundred base pair fragments and then amplify those specific regions to create the library. We ligate adapters on the ends of the fragments, and then tailed primers are used to add primer sites or patient-specific sequences to each sample library. We select specific gene targets with probes, add index sequences to the ends of the fragments, and then there's reversible dye terminator sequencing and ion conductance sequencing. Sequence analysis, image per data processing, sequence quality versus base calling. There are standards that are set by Clinical Laboratory Standards Institute that have particular scores that are necessary in order for sequencing to be valid. It depends on, or the coverage or read depth depends on the amount of template and the target size. Whole genome, 30 to 40 times coverage. Exome, 50 to 100 times coverage. 500, greater than 500 times is required for a rare variant in a mixed population. Cost is a limiting factor, so of course we could do it, it's just at what cost? Finding variants, the alignment of data in comparison to reference sequences. Reference sequence may have minor alleles. The most probable genome, theoretical probability of the variant call being correct. Properly aligned, non-repetitive, representative of the sample. Filtering variants, examine population frequency, ethnic reference genomes, thousands of genomes, and biological significance or the effect on the protein. Annotating variants, clinical significance, variant pathogenicity, reference sequence used to refine profiles. For example, a couple are listed here. Reference to unaffected family members or non-malignant tissue. Use human genome organization nomenclature. The significance of a variant is affected by incomplete assessment of structural variants, the lack of accurate haplotype data, variable expressivity, penetrance, environmental interactions and epigenetics, heritability, rare mutations of unknown significance, locus and allelic variability. NGS sequences are not always complete. Archiving. The extent of data to archive has not been established. Average NGS generates terabytes of raw data. Image files require the most storage capacity. Maintain different types of post-alignment files. So there's the FASTQ format and FASTA format. NGS in the medical laboratory, gene panels, targeted gene panels, 10 to 500 genes are sequenced, 16S rRNA, HIV genotypes, mitochondrial DNA sequencing, and whole genome sequencing. And the summary is provided.